Transporting substances across the membrane. All right. So we started talking about passive transport and active transport briefly. And now we're going to get into them in detail. So passive, you're going to have simple diffusion, you're going to have facilitated diffusion, and then active transport requires energy. And we're going to talk about the different forms of active transport. Diffusion, very simple. Moves from a high to a low. Think about riding a bicycle. If you're on a bicycle, and I'm a terrible artist, but anyways, they're on a bike, right? And the bike's at the top of a hill, and they want to ride down that hill. No pedaling, no energy is required. No energy. Why? Because those wheels will just roll down the hill. You're not needed to require to use any energy. And then when it becomes flat, you've reached equilibrium, you're dead. So things go from a high concentration. I should say molecules are like humans. They do not like to be smushed and touching one another. Molecules randomly are moving and they don't want to bump into one another. And so they are going to go to an area where there's less molecules because they like more space around them, just like humans. So diffusion is this passive process. We all move towards an area where there's less people, more space, from a high to a low, no energy required. That's diffusion. And so what determines diffusion across the plasma membrane? Well, the concentration gradient. Now, concentration gradients store potential energy. Again, think about the proton concentration gradient in inner mitochondrial membrane. This right here, created by the electron transport chain and oxidative phosphorylation, the higher that concentration gradient, the higher it is between the inner membrane and the inside of the mitochondria, the more energy available to create ATP. Concentration gradients are potential energy. Also, what determines diffusion across the membrane? Solubility. Nonpolar lipid molecules and gases, oxygen, carbon dioxide, are able to go through lipid bilayer. They can go through cell membranes. So that's the solubility. Presence of membrane ion channels. Facilitated diffusion. This is for facilitated diffusion. These ion channels are not always embedded in the cell membrane. The example I gave you with the GLUT4 channels, they're not always embedded in the cell membrane. They need insulin to facilitate their insertion in the membrane. And so those channels are not always there for facilitated diffusion. Diabetes is an issue with getting these GLUT4 channels into the cell membrane because they're not always present in the ion channels. And so presence of membrane ion channels does factor into the ability for things to diffuse. Even if it doesn't require energy, it requires these channels to be in the membrane. And then also, what is the membrane potential? What's the electrochemical gradient? What's the concentration gradient? So the factors that affect the rate or speed, how fast things diffuse across the membrane. Do you think it matters how fast things diffuse? Absolutely. Physiology is based in chemical reactions. Things have to be available and present for these reactions to occur. And if there's a supply chain breakdown, the cell's not getting what it needs, diffusion rate's too slow, that's the supply chain breakdown. Cell's going to die waiting for these supplies to arrive. So things that affect the rate's really important. The speed of diffusion is the magnitude of the driving force. How big are the concentration gradients and electrical gradients, if there is an electrical gradient? Number 2, surface area. Bigger the area to diffuse across, guess what? Faster molecules or solutes can diffuse or move, right? More lanes, you can go faster. And then membrane permeability. And this could be anything from, how many channels are available to move a substance across the membrane, right? That controls the permeability. So there are other factors, and we'll go over those. So diffusion rate versus distance. Listen, diffusion only works for small distances in general, but the bigger that distance gets, the longer it's going to take to diffuse across. And so diffusion is really limited by distance. That's why we have a circulatory system, because diffusion doesn't work for very long distances. We need the blood to carry stuff large distances. But if a substance has to cross a long distance, it's really going to take a while for diffusion to happen, and it's going to slow things down. So this example, cystic fibrosis, there's really an increase in the amount of mucus, right? And so if you have your mucus, the membrane has a bunch of mucus on it, and it's going to increase the distance. I'll do some red so you can see. So there's now an increase in distance for oxygen to travel across the membrane, respiratory membrane. And you have to remember, oxygen's coming from the inside of the alveoli of the lungs. It's got to diffuse across the respiratory membrane into the blood. Blood's moving. It doesn't stop and wait for the oxygen to load onto it. So if the oxygen has to travel too far, that red blood cell's already passed it by, and that red blood cell has not loaded up 100% with oxygen. So you're going to have a lower O2 saturation in someone with cystic fibrosis. Why? Because the mucous membrane increased the distance. The oxygen took too long to travel across that distance. It decreased the diffusion rate. Red blood cells went by. Hemoglobin didn't get loaded up with oxygen, took too long. And so you have a lower O2 saturation. We saw this with COVID. So there was a cytokine storm. There was an increase in inflammation of the respiratory membranes. Oxygen was taken too long to diffuse across because of that increased thickness or distance, and people were having low oxygen saturation levels. So magnitude of molecular movement or diffusion, we call this the flux. Flux just means like bulk movement of things, is dependent also on the temperature. If you kind of increase the temperature of molecules, they tend to move a little bit faster, right? Dancing around, it's warmer. We tend to want to move a little bit more, oxygen or molecules move a little bit more when they're faster. When it's really, really, really cold, oxygen molecules slow down and they don't diffuse as fast. So temperature matters. How big they are, solar molecules move faster. So as you increase the size of the molecule, it is going to decrease the flux. Surface area, you increase the surface area, you provide more area for molecules to diffuse across, it's going to be faster. And then the medium, things move faster in air than they do water, because water is a denser medium, the end molecules are going to move slower. So the more fluid there is, the slower the diffusion rate. Membranes are barriers, and that in itself is going to slow down molecules from moving across their surface, and specifically polar molecules, because they need a protein or ion channel. And so the number of these, are going to affect the rate or speed of diffusion. So the more channels you have, the faster the diffusion. Nonpolar, they're going to go right through the cell membrane. So that's not a factor for them. Oxygen, carbon dioxide, any kind of gases, and then any lipid-based molecules. So we do have some hormones that are nonpolar. We say they're lipophilic, they are lipid-loving, they are hydrophobic, they do not like water, are going to pass through, but polar molecules are lipophobic. It's an L. But hydrophilic, they need carriers. And so this is mediated transport. You need carrier proteins to bring these molecules into and out of the cell. This is an example of facilitated diffusion. So you have a solute, it's going to bind, that's going to change the carrier's conformation or shape to release it on the inside of the cell, and then it's going to go back to being the initial conformation, so it can take another particle. Now, these carrier proteins can also have different speeds of how quickly they get a substance across the cell membrane. So characteristics of these carriers, they're embedded in the membrane, so they're transmembrane proteins. They have a binding site for specific particle. They are specific to a solute, a specific solute. So they don't work for just anyone. They're very specific to a specific solute. Binding occurs on one side, transformation happens, solute's released on the opposite side, and it's going to go back to its original shape so it can start transporting all over again. The factors that are affecting facilitated diffusion rates as how fast did that transporter work to transport the substance across the membrane, and then the number of these transporters embedded in the membrane, and then the concentration gradient of the solute or substance. If you have a really high concentration gradient of the solute, guess what? That's going to affect the rate, just because there's more of them. Now, you see in this graph here, it plateaus. So this is the concentration gradient. As the concentration gradient goes up, the speed increases, right? Because net flux is really speed. But it plateaus here. It means it gets to this speed, and that is it. That's as fast as it's going to go. Why does it plateau? Well, that's when you hit full saturation of the number of carriers available. So concentration gradients only work until you hit the maximum number of carriers being used, and it can not go faster than that. GLUT4, again, these protein channels, they're transcription and translation of proteins, right? Transcription in the nucleus into a messenger RNA. Messenger RNA comes out and gets translated into a GLUT4 protein channel. It's finished up in the rough endoplasmic reticulum. And then they are packaged in vesicles, thanks to the Golgi apparatus. Those vesicles are waiting around with these GLUT4 channels, just waiting to be inserted in the membrane. And what's going to trigger this? You guessed it, insulin, but also exercise. This also does help. And so that's why exercise is super important for people with diabetes, because this is also a stimulant to release those GLUT4 channels into the cell membrane. And when they do, you're going to increase the number of carriers available, and you're going to increase diffusion of glucose into cell. Yay. All right. Characteristics of these channels, they're transmembrane, they function like a pore. These are really ion channels. A lot of times they're gated and they're specific for specific ions. There are aquaporins, those are specific for water. But ion channels, there's really three forms. There's leak channels, they're always open. There are gated channels, not always open, right? That's the gate. And then bidirectional. And again, the factors that are going to affect, right? Transport rate, number of channels, and are they open or closed? We're really going to get into these in the nervous system, but right now I'm just previewing it. The types of gated channels, right? So if we go back and we're talking about gated channels here, there are ligand gated, any type of protein that's going to bind to that gate and open the channel or gate. So ligands can open or close gates, mostly open. Voltage, so a change in voltage opens these up, or mechanically gated, meaning if you mechanically push or move, they're going to open. And so they allow ions in. So we will really be talking about these a lot in the nervous system. Active transport, it is non-spontaneous. It does require energy, and ATP is the energy source that it requires. It involves a pump, and it's really going uphill or regardless of a concentration gradient. So it can go up a concentration gradient. There's two types of active transport. There is primary active transport. This directly uses ATP energy. So remember, ATP stores potential energy. That energy is going to get released to then pump those sodiums out and then pump those potassium in. So it's called the sodium potassium pump. It's pumping both sodium and potassium. There's also secondary active transport, uses a concentration gradient developed by primary active transport. And so here you have a sodium potassium pump. It's pumping 3 sodium out for every 2 potassium in. But what it's doing is actively transporting sodium out, so you have a low sodium concentration gradient here. And if this is the inside of, say, your small intestines, and you had some yummy chips and there's sodium, it's going to be high concentration gradient out here in the inside of your small intestine and low inside the epithelial cell. And so now we have this concentration gradient where sodium has a drive to come down. And anytime you have things coming from a high to a low, there's a release of energy. And that energy is being used to transport glucose across the cell from a low concentration, and because it's all coming in here, it's a higher concentration. So this is secondary active transport of glucose because it's using this concentration gradient made by primary active transport. So we will go over this concept a number of times, don't worry. So ATP is pumped. uses energy. There's only so many of these, and so when they're 100% saturated, that's as fast as the transport can happen. This is primary active transport. These ATPase pumps. Why? Because the enzyme is binding the ATP, taking the energy out of that inorganic phosphate and using that energy. So uses energy stored in ATP to actively push substance across membrane, because you cannot rely on the high to low thing. There's no concentration gradient. And so the transporter has this enzyme, and transporter binds ATP. That's going to change the affinity for the sodium. So sodium has a higher affinity, a higher attraction is going to bind. That inorganic phosphate phosphorylizes and puts the energy into the transporter. ADP is released. That's going to cause the conformation to change, and sodium is going to lose affinity and be released. New affinity is for potassium, so it's now attracted to the transporter and it binds. And then the binding of the potassium causes the inorganic phosphate to dephosphorylize, which changes the affinity for potassium, and it's going to lose its affinity for potassium and release it in the cytosolic or intracellular side. And so is this is kind of how I like to remember how these physiological processes go. I always like to box it out. Like, what's the first thing that happens? Okay, this is number one. And what does that cause? And then I write it out. Causes number two to happen. And then number three to happen. And then number four to happen. And so when you're trying to study these processes, it's really great to break it down into these, chunks of this happens, then this happens, then this happens, and this happens. And these transporters, they'll just keep going as long as you have ATP and the need to transport sodium out and potassium into the cell. Secondary active transport is using primary active transport gradient that is created. And I really wrote that kind of poorly. It uses the gradient created by primary active transport. And so diffusion results from a concentration gradient that is created. So an example that I showed you before is the sodium link glucose pump. It's secondary active transport. Why? Because you had primary active transport of sodium out of cell. That created a concentration gradient for sodium, which is potential energy. And now glucose is linked with that pump to, as sodium ions just flow naturally down their concentration gradient, it's releasing energy that is harnessed by glucose to also go into the cell. So that is a secondary active transport. It's using an electrochemical gradient or a concentration gradient as its energy source, and that was created by primary active transport. So you could say it used stored electrochemical gradient, and this creation of this gradient is dependent on primary active transport occurring. If primary active transport doesn't occur, there is no secondary active transport. And so it relies on secondary active transport. That is to say, if you have no energy, no primary active transport happens, therefore no secondary active transport will happen. Secondary active transport can be in the same direction. It can also be in opposite directions. Intracellular fluid and extracellular fluid are different because of this primary active transport. And so it really does matter. We will talk about this throughout the entire semester. Last thing we really need to get down into the weeds about is osmosis, and that is the direction water moves. It's always passive, doesn't require energy, not really affected by membrane potential. It's really kind of driven by a solute concentration gradient. That is to say, it does depend on the number of channels or aquaporins available, right? So you got to have those available for water to move. But it also, if you have a semi-permeable membrane in this water, and these are solutes, and so you have 3 solutes here, and you have 3, 6, 9, 10 solutes here. This is a higher solute concentration gradient than this side, right? So we have a higher solute concentration gradient here and lower there. This means the solutes are almost touching. How dare they? And that's a high concentration. Solutes don't like that. So guess what? Water is going to move this way to separate those solute molecules so they're the same distance on both sides, right? They want to spread it out so they're that same distance on both sides. And so you have a high concentration and a low concentration. Water moves towards the higher solute concentration. So water is going to move, and you notice then these distances are going to be equal on both sides. Water moves up the solute concentration gradient to make the concentration of solutes in a given solution or given solvent the same. So you have equal concentrations on both sides. This is different. That's osmosis. Osmosis is the way water moves. Water moves up a solute concentration gradient. Osmolarity is the total solute concentration in a solution. So the osmolarity is how many solutes are in a given solvent. Because solute particles displace water, they're going to be kind of pushing that water around. And I want you to keep this mantra: water moves via osmosis down the water concentration gradient, but up the solute concentration gradient. The osmolarity is about 300 milliosmoles. That just means that's how many particles are in a given solution. These particles create a pressure on the water. It's osmotic pressure. And so as the solute concentration goes up, the osmolarity is going to go up. Maybe I should do this in black, right? You can see it. It's an up arrow, and the osmotic pressure is going to go up. So solutes create osmotic pressure in a solvent or fluid. And so water is going to move away from the osmotic or down the osmotic pressure gradient. And it's going to go towards the solutes. So it's moving up the solute concentration gradient. I guess the way to say it is water flows up its osmotic pressure gradient or the solute concentration gradient. Why? Because those molecules pull the water towards them. And that's that osmotic pressure. So if it's 300 millimoles on this side and 500 here, water's going to move up. And so water moves up the osmotic pressure gradient. If you have a membrane and there's solutions on both sides, isoosmotic would mean it's the same pressure on both sides. So, right, it'd be 300 on one side and 300 on. Hyperosmotic means one is higher than 300, right? And hypo would be below. Sorry about that. when we talk about tonicity, we're really talking about the solution or the osmolarity or the amount inside the cell. It's really to predict how water will move. And so you can have an isotonic solution. Isotonic solution is the same concentration as the intracellular solution. And let me denote this. Tonic solution is relative to the inside or intracellular fluid. So if it's isotonic, it's the same as the inside of the cell. If it's hypotonic, it means it has less concentration outside than inside. And if it's hypertonic, it's going to be higher than inside the cell. So it's a higher concentration in the extracellular. So why we talk about this? Because in physiology and medicine, people get intravenous fluids, and you really want to give someone isotonic fluid. So if you give them hypo or hypertonic, the cell is going to increase in fluid size or decrease in fluid size. And so isotonic would be what we normally give people, right? Hypertonic solution would be we give them a solution that is going to be higher. And so fluid is going to move out of the cell to where there's a higher concentration than lower. And so this would be hypertonic. If it was lower, right, this would be hypotonic. And I guess the way you could think about it is hypotonic makes the cell increase in size to become an O. So fluid is going to move into the cell because it's a higher concentration inside the cell, and that fluid's going to move into the cell to separate the molecules, right? So they separate the same distance. If it's hyper, it's really highly concentrated, what's going to happen here is fluid is going to move out of the cell, and the cell is going to shrink in size. And so that's the effect of the tonicity of a solution. All right, last thing. Transport of big molecules require vesicles to help them across the cell membrane. Can't rely on protein channels or anything like that. And so these vesicles, if it's bringing something inside the cell, endo means inside. And so endocytosis, cytosis is cell. It's bringing something inside the cell. Exo, meaning to exit the cell. It's bringing things outside of cell, or bringing stuff to the outside of the cell. I don't know, however you want to put it. So plasma membranes are very fluid. They can form a vesicle around something and become this is a membrane. It's from the cell membrane. And it just encloses around some large particles and brings it into the cell. That's endocytosis. If you have something stored in a vesicle, stored in membrane vesicle, and you need to get it out, so say this is insulin, right? You're going to exocytose it. You're going to bring it out of the cell. So movement of things in and out of the cell requiring vesicles. This is active transport. It does require energy, and vesicles have to form to bring it in or in the case of exocytosis, for it to be released. And so they are using membranes. Phagocytosis is specific for bacteria and viruses. And so the cell membrane will engulf the bacterium and bring it into the cell, form a phagosome, and that will merge with the lysosome, which has enzymes, and it's going to kill the bacteria or virus. So that's phagocytosis or phagocytosis. Pinocytosis is cell drinking. That just means it's taking a bunch of liquid and the particles inside the liquid and bringing it into the cells, cells drinking it, forming these vesicles. There's receptor mediated, meaning when a ligand binds to a receptor, it's going to cause a vesicle to then be created. And so that is a specific type of endocytosis. Exocytosis really just cell membrane merges. The vesicle cell membrane is going to merge with the cell membrane, and release the contents. Now, apical and lumen. When we talk about epithelium, hopefully you remembered epithelial cells from anatomy. There is a basolateral side and there is the apical side, which faces the outside or inside of a tube or lumen. That was supposed to be lumen. And so this would be the apical side and then the basolateral side. Important to understand that because say this is the small intestines. What foodstuffs you have is going to be facing the apical side of the columnar epithelium, and then the basolateral membrane is going to be connected to connective tissue. There's interstitial fluid and then the bloodstream underneath it. And so technically when things are in the lumen, they're not inside your body, has to go through the epithelium to get inside the body. And so this is where we get into transport across epithelium. And a lot of times this relies on primary active transport to transport substances, and then some of them get secondarily active transported across. And so things can get into the body via primary active transport and secondary active transport. And so really this is just reviewing those two. And this is just the example that was previously given about secondary active transport, right? So this is second and this is first. So you can review that primary and secondary active transport. And then water is going to require aquaporins, and the osmotic pressure of the interstitial fluid is going to determine which way the water is going to move, right? So if it's going to follow solutes, so water always moves up the osmotic pressure gradient, or another way to say is water moves up a solute concentration gradient. I always like to say water follows solutes. Transcytosis, just going from one side of the cell to the other, can use vesicles to do that. And that is the end.